All right, guys, so welcome to class. Here we are on Wednesday, March 9th. So what we're going to do in lecture today is we're going to finish up chapter 9, which is your chapter on senses, and we'll probably start chapter 10. And next Monday, and I will post an announcement about this with some study tips. On Monday, March 14th, will be your lecture exam 3, which will be on chapters 7 to 9. So next lecture exam will be on Monday, chapters 7 to 9. Chapters 7, 8, and 9 include muscular, nervous, and senses. And I will post some study tips for you either tonight or tomorrow about this lecture exam. But again, as you can see here, we're about a week behind, but I'm not super concerned because we'll have time to make it up. So a week from today is your lecture exam, March 14th, or not a week from today, on Monday is your next lecture exam, which is March 14th. On chapters 7 to 9, I will post an announcement about that with some study tips. We are kind of following the same schedule as lab. So if you have lab with me today on Wednesdays, we'll be going over kind of our first nervous system lab with a sheep brain dissection or looking at a video of one. So again, today we're going to finish up chapters 9. We will start chapter 10 on your endocrine system, but just keep in mind your test on Monday, the 14th, will just cover chapters 7 to 9. But I just want to get started on chapter 10 if we can. So with that, I'm going to go ahead and start talking. We'll finish up our senses. We were in the eye. I was hoping to get through the eye last time, but we didn't quite get it. Let's talk a little bit about how action potentials get to the brain. So this is the neuronal pathway for vision of the eye itself. So vision is one of your special senses. And the neuronal pathway for vision uses your optic nerve, which will leave your eye and exits the orbit through the optic foramen to enter the cranial cavity. Then the two optic nerves that come from your eyes kind of connect and cross over at the optic chiasm place. And the optic tracts will be the route of those ganglion axons up to the brain. So here we have a look at the optic nerves, and the place where they connect or cross is the optic chiasm. And then the optic tracts will take that information from the eye into your thalamus, and then it will be projected back to the occipital lobe, where you have your visual cortex center. So this is just a look at the visual pathway. You don't need to know all the steps of this, but I like to talk through this pathway just to show you that, from your right and left eye, you have different vision fields, and you can see those vision fields shaded different colors, and then follow the shading of that color up the specific pathway. You'll notice that part of your left vision field will get projected and kind of processed on the right side of the brain, and part of your right vision field will cross over and get processed on the left side of the brain. We have what we call the binocular vision field, where both eyes kind of focus on the same things. And then the right and left monocular vision field is more of your peripheral vision that your eyes can see in the periphery. So that's the visual pathway. The action potentials from the retina will get passed through optic nerves. They'll cross at the optic chiasm, follow optic tracts to the thalamus, to optic radiations, and then they'll eventually be sent to the visual cortex in each hemisphere of your occipital lobe, where all the visions and light that's coming into your eye will be processed. So that is the visual pathway. Some visual defects, they're a little tricky to understand. Myopia, let's highlight this one because the myopia is called nearsightedness. And this is when you cannot see things far away. So if you weren't able to see the whiteboard or chalkboard in 5th or 6th grade, you had myopia. So it's also known as nearsightedness, but it means you cannot see things far away. And that's because the image is focused in front of the retina. Hyperopia is farsightedness. This means when you cannot see things that are near or close up, and the image is focused behind the retina. And presbyopia is when the lens becomes less elastic and reading glasses will be required. So those are kind of the 3 different types of visual defects. We also have astigmatism, where the curvature of the lens is irregular, and you usually need glasses or contacts to correct that. If you already have myopia and you already wear glasses or contacts, sometimes they will test you for astigmatism, and they can put like a corrective lens for astigmatism right kind of with your normal prescription lens if you need a prescription already. Color blindness is absence or deficiency of one or more of the cones. Remember, the cones are blue, green, and red, and you can be colorblind for just one of those cones if one of them is deficient, or you can be colorblind for all of them. And primarily, for some reason, color blindness affects males, and I don't think we really know why that is. Glaucoma is increased pressure in the eye, and that can lead to blindness by putting increased pressure in the eye. Here are very popular charts that are used to determine color blindness, and if we were in lab in person, we would probably give you guys these charts to look at. But what they're doing is they're testing, for example, they're testing for red-green color blindness here and red-blue color blindness on the right. And you should be able to see numbers that are different colored circles. So this is showing the number 74. And if you're not able to see that, you might be colorblind for the green cone. And this on the right is showing the number 5. And if you can't see it, you might be colorblind for the blue cone. Then we'll go into the ear. So this will be equilibrium and balance and hearing. So your organs of hearing and balance are located in your ears. And we divide the ear into these 3 areas, the external, the middle, and the inner ear. And we'll kind of go through each section of the ear separately here. The external ear will extend from the outside of your head to the eardrum, which is your tympanic membrane. The auricle is the fleshy part on the outside. The external auditory meatus is the canal that leads to the eardrum. And the tympanic membrane is the actual correct term for eardrum. It's a very thin membrane that separates the external and middle ears from each other. The middle ear is completely filled with air, and it's filled with these 3 tiny ossicles that are so tiny that they don't even fill up the head of a penny. So if you have a penny next to you, these 3 tiny bones in your middle ear won't even fill up the space of a penny. The malleus is the hammer, and this is the bone that's attached to the tympanic membrane. So that's your first ossicle. The incus, the anvil, is the bone that connects the malleus to the stapes. And the stapes looks like a little stirrup, kind of what you would see on a saddle if you've ever ridden a horse. And this is the bone that is located at the base of a very thin kind of membrane or connection, what we call the oval window. And we'll kind of go through what that is. The oval window then is just a thin membrane that separates the middle and inner ear. And also in the middle ear is what we call the eustachian or auditory tube. And it will actually open into your pharynx, kind of in the back of your nasal cavity. And what this tube will help do is it helps to equalize air pressure between the outside air and the middle ear. So if you're up at very high altitudes in an airplane, or you're just up in the mountains, the eustachian tube helps to equalize pressure from the middle ear. And the popping sound that you hear is actually the tympanic membrane or the eardrum kind of popping back into place when pressure is equalized. The inner ear then is different in that it's a set of fluid-filled chambers. So we have a bony labyrinth, which are bony, kind of bone tunnels that are filled with fluid. And these are the 3 regions. We have the cochlea, the vestibule, and the semicircular canals. And then the membranous labyrinth is inside those bones, and it's filled with endolymph, which is a type of fluid. So that sound vibrations that are coming into your ear actually get passed on to create kind of waves through this fluid in the inner ear. The endolymph is a clear fluid in the membranous labyrinth, and the perilymph is a type of fluid between the membranous and bony labyrinth. Your cochlea is the specialized organ where hearing takes place, and it looks like a snail shell-shaped structure. And eventually we'll get to pictures here. The scala vestibuli is one of the kind of chambers in the cochlea that's filled with perilymph, a type of fluid. The scala tympani is in the cochlea filled with perilymph. And the cochlear duct is also in the cochlea filled with endolymph, which are just different types of fluid. The spiral organ is in the cochlear duct, and the spiral organ is specifically where we'll have hair cells that will bend into the tectorial membrane. And the vibration of those hair cells when they bend will be attached to a sensory neuron that will produce an action potential. I promise a picture soon. The vestibular membrane is the wall of the membranous labyrinth that lines the scala vestibuli, and the basilar membrane is the wall of the membranous labyrinth that lines the scala tympani. So let's take a break from me talking and look at some pictures. So here we have the external ear. It's from the outside where your auricle is, the fleshy part, through the external auditory canal. And then it ends at the tympanic membrane, which is here, and this is where your eardrum is. The middle ear then consists of an air-filled kind of chamber where you'll have these three ossicles, the malleus, incus, and stapes. And you'll see the auditory tube is the tube that will exit from the middle ear and enter into the pharynx of the nasal cavity to help equalize pressure in the middle ear. So these external ear and middle ear are all filled with air. And what happens is sound waves, vibrations from sound waves travel into the canal. They vibrate against the tympanic membrane. And then those vibrations get sent through these teeny tiny ossicle bones. Those ossicles will then send those vibrations into the inner ear. But remember the inner ear, it's composed of the semicircular canals, the vestibule, and the cochlea. These structures are all filled with fluid. So those sound waves that are vibrating through the tympanic membrane, the ossicles, those sound waves actually get changed into kind of waves that travel through the fluid of all of these inner ear chambers. So that is the inner ear. You can see the vestibular cochlear nerve, which will be one of your cranial nerves that will pick up these action potentials that are coming from the inner ear structures. So then we'll look at the inner ear itself. And first we'll look at the cochlea. So here we have the cochlea. And if we kind of take a cross section through one of these kind of tubes of the cochlea, we'll see that your cochlea is divided into different chambers. We have the scala vestibuli chamber up here. Below it, we have the scala tympani chamber, and these are all filled with different types of fluid. And then we have the cochlear duct chamber, also filled with fluid in between the two. And this cochlear duct chamber is then what we're kind of focusing on in this bottom picture. Because within this cochlear duct chamber, we have what we call the spiral organ. And the spiral organ consists of supporting cells with hair cells. And I'm just going to zoom in on one of these hair cells. You can see here that these hair cells are embedded or reach up and into this kind of tectorial membrane structure. And they are covered in microvilli. And at the bottom, they have a nerve ending attached to them. So what happens is these kind of waves that are traveling through these chambers will bend the hair cells, and the bending of those hair cells will be picked up by these nerve endings. So the mechanical bending of the hair cells will then be turned into an action potential that gets picked up by nerve endings and then sent out to the cochlear nerve through to the brain. Here's a very simplified view of the hearing process. So sound waves or sound will travel in waves through the air, and it's funneled into the ear by the auricle. The auricle through the external auditory meatus will then, these sound waves will then travel through the tympanic membrane. The tympanic membrane will vibrate and the sound is amplified, the malleus, incus, and stapes, which transmit sound to that oval window. And the oval window is what will produce the waves and then the fluid of the cochlea. The vibrations or waves in that fluid, the perilymph, will cause the vestibular membrane and the endolymph to all vibrate. So you have kind of waves passing through this fluid in the cochlea, The endolymph will cause a displacement of the basilar membrane, and that movement of the basilar membrane is detected by those hair cells. And when those hair cells become bent, that causes an action potential to be created. And this is a great look at how these sound waves will vibrate from 1 to 2 to 3. Here's the oval window where the stapes is attached. The stapes kind of bangs against this oval window. So those vibrations get turned into waves that travel through the fluid-filled cochlea. And as those waves travel through these different chambers in the cochlea, they'll cause the bending of those hair cells, which get picked up by your cochlear nerve. These kind of sound waves that travel through the cochlea eventually need to kind of be released so that pressure doesn't get built up from all these sound waves in the cochlea. So we have what we call the round window, which is another just thin piece of membrane that helps to absorb or kind of get rid of the sound waves that travel through the cochlea. So eventually the waves that are traveling through the fluid-filled cochlea will exit out of the round window. So the round wave kind of absorbs to kind of get rid of and keep everything get back to resting. So that's kind of the hearing process of the ear, a very simplified look. The balance process of the ear is also called equilibrium. Static equilibrium is associated with the vestibule part, which we'll talk about, and this will help to evaluate the position of your head relative to gravity. So if you look down quickly or look up, or are in an elevator and are moving up and down, your vestibule will help with that static equilibrium. Dynamic equilibrium is associated with the semicircular canals, and this evaluates changes in direction and the rate of head movement. So this is more associated with rotational movement. Let's say you're a figure skater and do 100 spins really quickly in a row. Dynamic equilibrium will help your body to maintain its composure. So the balance itself, the vestibule, we'll talk about this. It's in the inner ear. The vestibule contains the utricle and the saccule. And the maculae are specialized patches of epithelium in the utricle and saccule that are surrounded by endolymph, and they will contain hair cells as well. Otoliths is kind of like a jelly-like substance that moves in response to gravity. They are attached to hair cell microvilli, which initiate an action potential. So a similar type things happens. We have hair cells kind of embedded within this gelatinous mass, and the gelatinous mass will move in relation to kind of gravity, your head moving up and down, and that will cause bending of these hair cells. And when those hair cells bend, that mechanical bending is picked up or turned into an action potential that gets picked up by the vestibular nerve and eventually taken up to the brain, so your brain can realize kind of what is happening in terms of balance. Here is the vestibule and kind of where the utricle and saccule are located within this vestibule portion. These three kind of canals, those are the semicircular canals that we'll go over next. So here's a look at the function of the vestibule in maintaining balance. So just really quickly, just looking down, your head being down, that movement, the force of gravity will force these otoliths to move the hair cells. And again, the movement of those hair cells is picked up by your brain, so your brain can determine how it is bending in response to gravity. Balance then is also kind of picked up by your semicircular canals. This is dynamic equilibrium, and this will sense movement in any direction. So think of rotational movement of your body or your head. We have similar structures, an ampulla, a Crista ampullaris, a cupula, which is the gelatinous mass containing the microvilli, and they will float, or it will float and it'll be displaced by any sort of movement. So here we have the semicircular canals within the ampulla. As you can see here, you see a cupula, and the cupula itself is this gelatinous mass that these hair cells are embedded in, called the Crista ampullaris. And again, when this cupula moves, usually due to rotational movement, that will bend the hair cells in your semicircular canals, and the bending of those hair cells is turned into an action potential, which is picked up by your brain so that your brain can process, oh, hey, we're rotating kind of fast here. I should probably compensate so we can maintain some sort of balance. So I talked about figure skating, think also of a gymnastic, maybe doing a flip off of a balance beam. You know, her rotational movement is causing the endolymph to move the cupula, and that will in turn bend the hair cells, which will be picked up by the Crista ampullaris as action potentials to then get sent up to the brain. Again, so that this gymnast will be able to kind of maintain her composure when she lands and not be all over the place or fall over. Okay, so that's the end of chapter 9, senses. If you joined us late, you will have a test on Monday. So I'm just going to pull up our schedule one more time for those of you who might have joined us late. So your lecture exam 3 on chapters 7 to 9 will be on Monday, which is March 14th. So we're just a couple days behind in lecture. Chapter 3 includes the muscle system, the nervous system, and the senses. And in lab, we're going to maintain our normal lab schedule. So this is what you'll have today with me in lab. What I'm going to do now is I just want to start the endocrine system a little bit. This is chapter 10. It won't be included on your next exam, but I just want to start it to try to get a head start. So let's get into your endocrine system here. And let me go ahead and exit out of the chapter 9 PowerPoint. Okay, so the endocrine system is one of my favorite systems because it includes little teeny tiny organs, hormones that virtually impact all parts of the body. So we'll just start this PowerPoint. Maybe we'll get through half of it. So this chapter is not included on your next lecture exam, but do look for an announcement posted with some study tips by this weekend for your next lecture exam too. Okay, so the endocrine system glands are shown here. And these are glands in the body that produce hormones that control many of your bodily functions, from metabolism, which turns food into the energy that your cells need, your sleep-wake cycles, your nervous system, skeletal system, reproductive system, levels of insulin and glucose in the body. So these are your endocrine system major glands. We have the pituitary gland, the pineal gland located in the brain, the thyroid gland with parathyroid glands on the posterior part of it, kind of embedded within. The thymus is a part of your endocrine system and lymphatic system. We'll talk about that. You have adrenal glands located on top of your kidneys. You have the pancreas endocrine system functions, and then you have ovaries and testes, which are called the gonads, and they will secrete different hormones as well. So what do these glands secrete, and how do these cells secrete their hormones? We'll talk a little bit about how cells can secrete hormones that are being formed within them. Cells can secrete substances by an autocrine secretion, which is released by cells that has a very local effect on the same cell type that it's being secreted from. And an example of this is an eicosanoid. And we'll talk about what that is. Paracrine secretion is released by cells that have an effect on other cell types that are in close proximity. And an example of this is somatostatin. And we'll talk about what that is. That's a growth hormone, inhibiting hormone. Neurotransmitter and neuromodulators are secreted by nerve cells to have nervous system functions. And then hormones and neurohormones are secreted directly into the blood. They will bind to specific receptor sites on a specific type of cell. And an example of hormones are epinephrine, which gets your body ready with adrenaline, and insulin, which lowers your blood glucose levels. The endocrine system functions are very diverse and very vast. And again, these functions are controlled by teeny tiny chemical messengers called hormones. And that's what makes the endocrine system so interesting. It controls your metabolism, which is the ability for your cells to turn food breakdown products like glucose into energy. Your endocrine system function controls food intake, so how hungry you are and how your body digests that food. Your endocrine system controls tissue development, ion regulation, and water balance in the body. It also functions to control heart rate and blood pressure. It controls blood glucose levels and other nutrients. Have you ever heard of insulin? Do you know of anyone who's diabetic? We'll talk about that. Your endocrine system controls reproductive functions, the development of secondary sexual characteristics, the start of puberty, maintaining reproductive functions. Those are all a part of your endocrine system and their hormones. Uterine contractions, giving birth is an endocrine system function. Milk release, if any of you have nursed babies, you know what that feels like, as well as immune system regulation. So your endocrine system is very diverse. You can see how it has its hand in almost every part of the body's functions. Some characteristics of your endocrine system: it's composed of endocrine glands, which we kind of showed you a couple in that first picture, and specialized endocrine cells located throughout the body. So there are specialized endocrine cells in your digestive tract, or you might have an entire endocrine gland like your thyroid gland. Endocrine glands and cells will secrete minute, very small amounts of chemical messengers called hormones directly into the bloodstream. So that's what makes an endocrine gland unique, is that it secretes its messengers, hormones, directly into the bloodstream. And the blood acts as the highway of transportation for these hormones. Exocrine glands secrete their substances into a duct, which is like a tube that carries it. So endocrine system uses your blood to transport hormones, and exocrine glands, like sweat glands, will use ducts or tubes to transport their substances. Hormones will then travel through your general blood circulation, through your arteries and veins, to a specific target tissue or an effector that it wants to have an effect on. And it's very important to remember that the target tissues have very specific receptors for a specific hormone. So this means that hormones just can't go and kind of attach to any cell they want to. A hormone will only have an effect on the cell that has a receptor for it. So hormones will produce a very particular response in the target tissues that they're traveling to. Hormones can be what we call water-soluble or lipid-soluble. And remember that the cell membrane of our cells, the plasma membrane, is made of lipids. So if a hormone is lipid-soluble, it means it can get through the cell membrane because the cell membrane is made of lipids. And this will include your steroids and eicosanoids like luteinizing hormone, follicle-stimulating hormone, and androgens that all have to do with the gonads, the testes and ovaries. If you have a water-soluble hormone, this could be a protein, a peptide, an amino acid. These are the most common. Examples of these are growth hormone, antidiuretic, and prolactin. If it's a water-soluble hormone, it cannot enter into the cell because it can't get through the lipid membrane because it won't be able to dissolve through it. In order to control hormone release, the blood-borne chemicals can directly stimulate a release of a specific type of hormone. And these chemicals are referred to as humoral stimuli because they circulate in the blood. Humoral refers to body fluids, including blood, and hormone release can also be under neural control, meaning that your nervous system has a say in when these hormones are released. So for example, following an action potential, a neuron, a nerve cell, can release a neurotransmitter into a synapse that's connected to a cell that can produce a specific hormone. Hormone release can also be controlled by other hormones. And what that means is it occurs when a hormone is secreted that in turn will stimulate the secretion of another hormone. The same three types of stimuli, whether it's humoral, neural, or hormonal, can stimulate or inhibit a hormone release. So that's how we control when hormones are released. So for example, if you have low blood calcium levels, this is an example of humoral regulation. The low blood calcium levels are picked up by an endocrine cell when blood calcium is too low. That will stimulate parathyroid hormone release to basically tell osteoclasts to break down bone to release more blood calcium or calcium into the blood. So this is to try to increase blood calcium levels. If you have too high blood calcium levels, again, this is a humoral regulation. An endocrine cell can pick up when blood calcium levels are too high. There's no parathyroid hormone secretion, so that osteoclasts will not try to put more calcium into the blood, and that in turn will decrease our blood calcium levels. Neural regulation, this just shows how a neuron can be directly attached to an endocrine cell. So it will release a neurotransmitter across the synapse, which will cause the endocrine cell to release some sort of hormone that will then travel into the bloodstream to reach its target. So that's endocrine cells under neural regulation. And then hormonal regulation, this is the one that we'll talk about a lot. This is when there's some neurons, usually in the hypothalamus. The hypothalamus will release a hormone that acts on your pituitary gland to release another hormone. That hormone acts on some sort of target endocrine cell that releases another hormone that finally acts on the target. So we kind of have this ladder of control or a chain of command where one endocrine gland releases a hormone that controls the release of hormone from another endocrine gland that eventually causes the release of the hormone that we want to reach our target cell. So that's called hormonal regulation as well. In order to inhibit hormone release, so many times we want hormones to be released. That's called a stimulatory effect. If we want to inhibit hormone release, humoral inhibition of hormone release generally involves the actions of a companion hormone, where one does the opposite effect of the other. And usually each of the companion hormones performs an opposite function. So for example, to raise your blood pressure, your adrenal cortex and your adrenal gland on top of your kidneys will secrete aldosterone in response to low blood pressure. But to lower blood pressure, your heart, atria, secrete a hormone called atrial natriuretic peptide. So we have aldosterone and atrial natriuretic peptide to work together to maintain the homeostasis of blood pressure. Neurons themselves will inhibit their targets just as often as they stimulate them. So we might be inhibiting hormone release as well. If your neurotransmitter is inhibitory, for example, the target endocrine gland will not secrete its hormone. And in control of the hormone release by other hormones, some hormones will be inhibitory hormones that reduce the release of the hormone being controlled. So for example, thyroid hormones from your thyroid gland can control their own blood levels by inhibiting their anterior pituitary tropic hormone. So this just describes how some hormone release can actually inhibit further release of hormones. For example, regulating your blood hormone levels, there's two major mechanisms that we talk about, and these are very important: negative feedback and positive feedback mechanisms that try to keep the levels of hormones always within the normal homeostatic range. And most of the hormones in our body are regulated by a negative feedback mechanism, which means that when the hormone secretion will be inhibited by the hormone itself, once blood levels of that hormone have reached a certain level. Some hormones, however, are regulated by what we call positive feedback. And that is when the levels of hormones will continue to rise and increase, and they'll never be shut off until an end goal is achieved. So here we have an example of negative feedback on the left. So here's negative feedback control by hormones. What this is showing is that when your target endocrine cell releases its hormone, when levels of this hormone get to be at an adequate or normal range in your blood, it tells kind of the first part, the anterior pituitary or your hypothalamus, to shut off production of that hormone. Because levels of this hormone, once they reach their normal homeostatic range, they will turn off this kind of production of the hormone in general. So that's the most common way that we regulate levels of hormone in the body. Whereas positive feedback mechanism, we have levels of hormones in the blood continue to tell the pituitary and your hypothalamus to continue secreting them. So there's no kind of turn off switch. A positive feedback mechanism, an example is with oxytocin stimulating the uterus to contract during childbirth. And if you've ever given birth to a child or been with someone who's done that, you know that contractions continue to get stronger and stronger. Levels of the oxytocin hormone, they don't shut off. They continue to get higher and higher to increase contractions until an end goal is achieved, which is the birth of the baby. So that's an example of a positive feedback. If you're a male and wondering how positive feedback is affected in you, another example of positive feedback is with platelets in your blood, which travel to a site of damage to your blood vessels. So let's say you accidentally cut a little piece of your finger when you're chopping vegetables. Platelets will continue to flood that area to try to form a clot to stop you from bleeding. So platelets coming to the area of damage is another example of a positive feedback loop. Hormone receptors, we talked a little bit about hormone receptors. A hormone can stimulate only the cells that have the receptor for that hormone. So the portion of each receptor molecule where a hormone will bind to a cell is called the receptor site. And the receptor site has specificity, which means that only one hormone can bind to it. Some hormones, like epinephrine, can bind to a family of receptors that are structurally similar, like epinephrine or norepinephrine, which are both adrenaline type hormones that get your body ready for activity. And this specificity is due to the molecular shape and chemical characteristic of the receptor itself. If we have a lipid-soluble hormone, which we talked about, they can bind to nuclear receptors due to their lipid solubility and small molecular size. They can easily pass through the cell membrane because remember, your cell membrane is made of lipids. But water-soluble hormones will bind to a receptor on the plasma membrane. They are polar and they will not dissolve through the lipid membrane. So they will never pass through the cell membrane. If it's a lipid-soluble hormone receptor, again, lipid-soluble hormones will pass through and eventually you'll have them pass into the nucleus. A nuclear receptor would be located in the cytoplasm, and hormones might even bind to nuclear receptors on the nucleus. And when that happens, this hormone receptor complex will interact with DNA to regulate specific gene transcription to tell that cell to create a specific protein in order to perform a function, for example. A water-soluble receptor, which we talked about again, water-soluble hormones cannot pass through the cell membrane. So they will just interact with cell membrane receptors. When hormones bind to receptors on the plasma membrane, it turns on kind of what we call an intracellular enzyme cascade that ultimately causes the response dictated by the hormone receptor interaction. So this just goes through, and I won't go too much further here, but this just talks a little bit about how each target tissue is very specific and responsive to a specific type of hormone. So here we have a hormone receptor that is very specific to a type of hormone that it can bind to. So we have a hormone 1 leaving the blood and binding to a specific type of hormone receptor that it can fit in. Hormone 2, for example, cannot bind to this receptor because it doesn't have the correct shape. So this is the target cell only for a hormone 1. This shows how lipid-soluble hormones are able to pass through the cell membrane and eventually into the nucleus to bind to receptors within the nucleus. And it will initiate a response from the cell by usually initiating some sort of transcription or creation of a protein of that cell. A water-soluble protein cannot get into the cell, so it will bind to a membrane-bound receptor. And it initiates kind of a sort of complex reaction called a G protein complex, cyclic AMP, to kind of make a cellular response that way. So it's just different ways that hormones whether they can get into the nucleus or whether they have to stay on the cell membrane. They'll both initiate a cellular response, but in different ways. So those are nuclear and membrane-bound receptors. The action of nuclear receptors, so this is for lipid-soluble hormones that can get into the nucleus, they will stimulate protein synthesis or the creation of new proteins. So when these lipid-soluble hormones get into the cell, bind to the receptors, they can now bind to hormone response elements on DNA, and this will start kind of the transcription process of creating a specific type of protein in order to initiate a cellular response. And you won't be asked too many specifics about this, but it's just, if you can remember that lipid-soluble hormones can get into the cell because they can pass through the cell membrane. So they will actually get all the way into the nucleus to bind with a nuclear receptor, and they will initiate a response by producing hormones, which is protein synthesis. So they'll interact with DNA to produce proteins that way. Membrane receptor action. So this is for water-soluble proteins that cannot get into the cell. They will interact with a membrane receptor by altering activity of G proteins or directly altering activity of intracellular membranes. If we activate a G protein or an intracellular enzyme, it will elicit some sort of response, including the production of molecules called second messengers. And a second messenger is a molecule that's produced inside a cell once a ligand binds to its membrane-bound receptor. And we're talking here about hormones. So once a hormone binds to a receptor on the membrane, we have second messengers that are produced. And a very important second messenger is called cyclic AMP. It will activate specific cellular processes inside the cell to respond to that hormone that's being bound to it. Many membrane-bound receptors will produce responses to activate G proteins, which consist of subunits because they bind to guanine. That's why they're called G proteins. And after many sequential actions, G proteins will interact with adenylate cyclase, and that will eventually convert or create our cyclic AMP, which is our second messenger that we want in order to bind to proteins to activate them. Protein kinases are in turn will then they will activate other enzymes, and depending on the type of enzyme, they can inhibit or decrease the activity of that enzyme. Phosphodiesterase is an intracellular enzyme that breaks down cyclic AMP and thus results in no further cell stimulation. So in general, what we're talking about here, kind of backing up, we talked about how lipid-soluble proteins or hormones can get into the nucleus of the cell to create a cellular response that way. Now we're talking about water-soluble hormones. Because they cannot get into the cell, how can a water-soluble hormone make a cell respond to it? And it does so by activating G proteins. And G proteins will go on and then activate cyclic AMP. And cyclic AMP will then go on to activate different protein kinases to get out a cellular response that way. Because remember, we're talking about hormones here, and hormones can do a wide variety of things in our body cells. But in order for our hormones to activate a cell and get a cell to respond to a hormone, it either needs to be lipid soluble, get into the nucleus and tell that cell to create new proteins, or if it's water soluble, it will bind to a membrane receptor and go through this process to initiate or elicit some sort of cellular response. Hormones themselves that can stimulate the synthesis of second messengers will act quickly and have what we call an amplification effect, where each receptor produces thousands of second messengers, leading to a cascade effect and ultimately amplification of that hormonal signal. When we amplify the signal, that hormone will activate many secondary messengers, each of which activates enzymes that produce an enormous amount of final product. And this signal amplification, the efficiency of this is virtually unparalleled in the body. So that's why teeny tiny hormones can have such a huge effect on bodily systems because of this signal amplification effect. So that's what simple signal amplification effect is. And this describes that signal amplification, showing how one hormone binding to one receptor on a cell membrane will activate G proteins that activate adenylyl cyclase, that activates cyclic AMP, that will go on and activate protein kinase enzyme. So this just shows how one protein can have this enormous effect on cellular production and cellular response to that hormone. Okay, so with that, I'm going to stop there and we'll finish this for your lecture exam on Monday. Again, you have a lecture exam on Monday. It will be on chapters 7 to 9, March 14th. I'll post kind of study tips. It will not include what we just went over on chapter 10. We'll just do your lecture exam on Monday. And then on Wednesday, which will be, what is that, March 16th, we'll finish up chapter 10 on your endocrine system. And then you guys have a week off with spring break. So thank you guys for listening. I'll stop the recording. You will